All right. Let's settle in and continue talking about synapses. Okay. So when we left off last time, thinking about the synapses, we were thinking about the different types of receptors that we see in chemical synapses specifically, remembering that electrical synapses are formed by gap junctions, cytoplasm to cytoplasm. Chemical synapses are ones with neurotransmitters. So we saw our ionotropic receptors, which we might also think of as channel-linked receptors, where a neurotransmitter binds into a protein that then acts as a channel for ions. And we thought about our metabotropic receptors, which have a slightly more complicated process that involves a G protein that's acting on the inside of the cell, doing stuff, but eventually our end result is the same. Some ion channel somewhere in the membrane opens, allowing ions to move in or out. Okay, so when we have our ion channels open, right, we are now going to see membrane potential changes, right? So we are looping all the way back now to our graded potentials, because that's essentially what we're doing with our neurotransmitters, right? We're telling the next neuron in the chain or an effector organ like a muscle or gland whether or not it should do something. And the way we do that is by changing its membrane potential, okay? So if we had an ionotropic receptor, a ligand-gated ion channel, we would see that ion channel open. If we open up a sodium or a calcium channel, both of those ions have a really strong electrochemical driving force into the cell. So if we open a sodium channel or a calcium channel, we're going to see that our graded potential in that cell that we're acting on is going to be excitatory. So excitatory, meaning that this is a depolarization, getting us closer to our threshold, getting us closer to actually sending off another action potential in this new cell now. Okay, so when we talk about excitatory postsynaptic potentials, or EPSPs, that's what we're talking about, all right? So we're talking about depolarization, possibly another action potential. Yeah. Now, if we open up a fluoride channel or a potassium channel, it's going to have the opposite effect. So our potassium wants to leave cells, right? So that would hyperpolarize. Chloride channels, the situation about exactly how they cause this hyperpolarization is a little more tricky. They can be kind of stabilizing. But in either case, this is going to inhibit an action potential in our postsynaptic neuron. So we call it an IPSP, or an inhibitory postsynaptic potential. But these are just new names applied to those same graded potentials that we talked about at the beginning of the unit. We just know more about them now. So when we have a neurotransmitter bind to a receptor, eventually we have some type of ion channel open. Ionotropic receptors, it's going to open immediately because that is part of the receptor. Metabotropic, we got some steps, but eventually it'll happen, okay? Giving us our EPSPs and our IPSPs. There are lots and lots of neurotransmitters in your brain, in your body. Their effects can be different depending on what cell they're talking to, what cell is receiving that signal, but we can make some generalizations. So our most common neurotransmitter that's related to excitatory postsynaptic potentials is a neurotransmitter called glutamate. So more glutamate tends to cause more action potentials, gets you closer to that threshold, tends to let sodium, calcium in. Our most common neurotransmitter for inhibiting our neurons is a neurotransmitter called GABA. Okay? So if you have GABA released from your presynaptic neuron, that's going to kind of suppress that postsynaptic neuron, make it less likely to fire off an action potential. So excitatory, more likely to produce action potentials. This means it's going to be a depolarization of some type. If we talk about something as inhibitory, this means it's less likely to produce an action potential. That means it's probably a hyperpolarization if we're thinking about potassium. We can also see some membrane stabilization happening if we think about chloride ions. So the only anion that we saw on our list of negative ions sometimes help it in a kind of stabilization process. And if you're stabilizing at -70, you're less likely to get the threshold up to -55. So that would still count as inhibitory. So here we see some examples of how this would work, right? Okay. So we're looking at our extracellular fluid up top, right? So this would be fluid in the synaptic cleft. So our presynaptic neuron would be somewhere over here, right? And we're looking at our postsynaptic neuron on the bottom. Okay. Okay. So here we can see that example of an ionotropic receptor or channel-linked receptor. Right, so here's our neurotransmitter coming in. So we can see that neurotransmitter binding. Okay. And we can see that the result is a slight surge in charge. Right? So we can see this little depolarization here, indicating to us that this overall effect was excitatory. So we can see that what actually moved through this channel was both sodium and potassium. So the reason this would be excitatory is that we have a larger force on sodium, right? We know that we have a really strong electrochemical driving force for sodium to move into the cell. So if we let sodium in, our overall effect is going to be a bit of a depolarization. On the other side, same thing, really. We're looking at a presynaptic neuron would be somewhere in this white space. Synaptic cleft is in here. Postsynaptic neuron is what we're focusing on. So we see our receptor. This time it's a metabotropic receptor, which we can identify because we have a G protein down here doing some stuff. In fact, this is our slower type of metabotropic receptor. We can see this longer action. Here we see a second messenger system. So we might expect this response in the cell to happen for a longer period of time, as well. Okay, and we can see that the effect here is another way that we could get excitation happening. So here, instead of opening a channel that allowed positive ions in, we closed a potassium channel. Okay. So when we close a potassium channel, right, this means that right at baseline, we know that we have a little bit of sodium and potassium leaking, right? We know that our membrane is 25 times more permeable to potassium at its baseline than it is to sodium, which is why we're down near that equilibrium potential for potassium. Right? That's why we're at -70. We talked about that when we talked about how that resting membrane potential develops. Okay? So what we've essentially done here is we've decreased that permeability to potassium. So now we don't have potassium leaving the cell as much, which means overall, we're going to have a slightly more positive membrane potential, slight bit of depolarization. So those would both be examples of ways we could get some excitation happening in our postsynaptic neuron, potentially help us get to threshold to fire off an action potential. On the inhibitory side, right, so we'd have the same kind of flow, right? So our presynaptic neuron, get that action potential to the axon terminal, trigger the voltage-gated calcium channels to open. Calcium rushes in. That triggers the vesicles holding our neurotransmitters, bind onto our membrane that's facing the synaptic cleft, secrete those neurotransmitters into the synaptic cleft, and that neurotransmitter is here in our space in this extracellular fluid, and it's coming in and binding to the receptor, which we're picking up in this image. Okay. So if we open up a potassium or a chloride channel, right, we have inhibition happening, right? So here we can see that inhibition as we see potassium leaving our postsynaptic neuron here. So as we remove positive charges, that cell becomes more negative, hyperpolarizing. And we can see that on our little voltmeter here, right? So we can see that hyperpolarization, which we're now thinking about as an inhibitory postsynaptic potential, because we're being specific that the specific cell type we're talking about is our postsynaptic neuron, and we're thinking about, is it going to give off an action potential or not? So for our chloride channels, the situation is a little more complicated. So one thing that can happen if you open a chloride channel is that the chloride can just directly move in. That version is pretty simple, right? So chloride is an anion, it's negative, that negative charge makes the cell more negative, kind of hyperpolarizing it. So inhibiting our postsynaptic neuron, so inhibitory postsynaptic potential. The other thing it can do, though, is to stabilize the membrane potential. So that's a little more complicated. I think we got, do we not have a picture of it? Okay. I don't have a picture of it here, but basically what happens there is that we do actively transport chloride ions. We haven't talked about it, right? But here you can see a chloride ion pump, right? So there's some energy being used to move chloride ions. And we can change a little bit about the behavior of chloride relative to the membrane and the other ions involved in the system. So sometimes chloride will kind of like move in and out following our cations and kind of canceling them out, essentially, is the basic idea of how that stabilization works. So when we're thinking about either our inhibitory or excitatory postsynaptic potentials, they just are graded potentials. Now we just have specific types. So if we were tracing how we end up with an excitatory postsynaptic potential, what happens here, right? We might be thinking about it's being signaled by its presynaptic neuron. So say we have a high frequency of action potential coming down that presynaptic neuron, right? So it's signaling pretty fast. Okay. A neuron that's getting lots of action potentials, going boom, 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 faster in sequence, is going to end up releasing more neurotransmitters. Right? Every time that signal gets to our axon terminal, that opens the voltage-gated calcium channel, calcium comes in, right? And that presence of calcium in general helps us release neurotransmitter, right? So if it's hanging around for a period of time, that's also going to increase neurotransmitter release, right? So when we have more action potentials in a presynaptic neuron, we have more neurotransmitter release into the synaptic cleft, which means there's more neurotransmitter that can bind to our receptors, whether they be ionotropic or metabotropic, right? And whether they eventually open or close an ion channel. So you'll want to spend some time practicing thinking about the ions, thinking through the steps of like, okay, we open a sodium channel, what happens? We close a sodium channel, what happens? We open a potassium channel, what happens? Close a potassium channel, what happens? But we've been talking about that as we talked through the behavior of those ions more generally in earlier parts of this unit. I have faith that if you think through those slowly, you can accurately get to whether we're going to have excitation, the depolarization, or inhibition, which for our purposes, if I'm asking you about inhibition, I'll ask you one of the hyperpolarizing versions. Okay. So if we have more neurotransmitter binding to receptors and we were thinking we want to get to a, let's do the hyperpolarization first, right? So in this case, we might, for example, open a potassium channel, right? Potassium would then leave the cell. This is an increase in potassium ion permeability, increase in potassium ion movement out of the cell, which would be a greater hyperpolarization. If we wanted to track one that's depolarizing, right, we might think about opening a sodium channel, increasing sodium permeability, more movement of sodium, it's going to result in sodium coming into the cell, depolarizing. If we do the reverse, let's think about sodium now. Let's say we close a sodium channel, we'd have even less sodium coming in, then less sodium coming in, might also get you to hyperpolarization. This is just a workflow for kind of thinking through how you get to inhibition or excitation. So for our excitatory synapses, a neurotransmitter is binding to a receptor. We'd open a sodium channel or a calcium channel. If we're thinking about channels that could close and cause excitation, right? We will sometimes see potassium channels closing, right? So we're thinking about sodium moving in, causing depolarization, or stopping potassium from moving out would also cause excitation because it's kind of not balancing out anymore that sodium slight leak that we have. Let's do an example. So for serotonin, it binds to a 5-HT3 channel receptor, allows sodium to cross the cell membrane. Is that going to be an excitatory postsynaptic potential or an inhibitory postsynaptic potential? Okay. Great. Excitatory? Yes. All right. Now, is that going to be a ionotropic or a metabotropic receptor? So let's actually go back to that description first. All right. So we have a 5-HT3 channel receptor. So is that ionotropic or metabotropic? Take another second or so. Great. Yes, our channel receptors are ionotropic receptors. So the serotonin and the 5-HT3 channel receptor are describing a system using an ionotropic receptor. Okay, cool. Here's my chloride picture. Okay. So what we see here is chloride equilibrium here when we have no active transport of chloride. Here's what we see as that chloride is kind of balancing out an excitatory signal here. So you can see that as these positive charges move in, we kind of have some chloride movement that is balancing it in the opposite direction, causing a kind of flat line here. Okay. So the next thing we're going to think about is how we handle multiple signals. Okay. So as a reminder, we got a bunch of types of ion channels. So we had leak channels responsible for keeping us at the resting membrane potential. They're always open, they're throughout our neuron. So those were our sodium and potassium channels, where we were saying there are 25 times more potassium channels. We talked about voltage gated channels as we talked about the action potential, and as we talked about the release of neurotransmitter from the axon terminal. And just now we've been talking about things like ligand gated channels. So like that would essentially be our ionotropic receptors would be an example of one of those that would open or close in response to a ligand like a neurotransmitter binding onto it. So that's going to be clustered in the parts of our cell that receive signals, so our dendrites and our cell bodies. All right. So as we're thinking about neural integration, we're thinking about like our big picture map of how communication is happening when we have more parts to the system. So we're going to think about all of these different things. We're going to be thinking about summation, which hopefully sounds a little familiar because we did a little bit of that as we were thinking about our graded potentials just on their own. And we're going to think about frequency coding, which is going to have to do with kind of the speed of action potentials, divergence and convergence, which is going to be about kind of directions of signals, let's say. And we're going to be making sure that we're focusing really on the axon hillock and whether or not it hits threshold, because that is what determines whether or not we have an action potential, which is our big picture question. Okay. So anytime that membrane of a neuron gets to threshold, that opens voltage gated sodium channels, so we have an action potential. We particularly care if that happens at the axon hillock, because that's what leads into the axon. So that's what would lead to communication, that wave of depolarization happening all the way down our axon to the axon terminal. If we don't get the threshold, doesn't matter how many signals we had come in, no action potentials happen. Okay. So we have connections that can look like this. Right, and this is why we're thinking about integration, neural integration. So putting together multiple signals. Okay. So sometimes we'll have it be the case that one single neuron, so here we see our presynaptic neuron up top, right? So it may be the case that as we come to the end of its axon, it splits out and has axon terminals acting on multiple different cells. So here we see 4 postsynaptic neurons, each getting that signal from the presynaptic neuron. This is what we refer to as divergence. So the signal is starting from one place and diverging, so splitting out, signaling to multiple different places. The opposite version of this is convergence. So when we have many different presynaptic neurons all converging on one postsynaptic neuron, that's then going to have to decide, is this enough to get us to threshold? Okay. So here in convergence, we're looking at the postsynaptic neuron, right? So we know our big question is, does the axon hillock depolarize to get to threshold to send off an action potential? But we're going to have to add up signals from each of our presynaptic neurons. So each of these is going to be releasing neurotransmitter, telling this postsynaptic neuron potentially different information. And in general, in your book, when you see something in green, that's indicating that it's excitatory. When you see something in red, that's indicating it's inhibitory. So in this image, they're showing that one postsynaptic neuron may be receiving both types of signals. You might be receiving some signals that are inhibitory, some that are excitatory, We kind of have to add them all together to figure out what's what. Okay. So adding them together is really what we talked about at the beginning when we said graded potentials can sum spatially, temporally. This is happening when we have convergence on a postsynaptic neuron as well. So we're just adding together the effects of graded potentials. So our IPSPs are inhibitory postsynaptic potentials, and our excitatory postsynaptic potentials, our EPSPs, are both just types of graded potentials, so they can add together in the ways we talked about before. Okay. So we can see temporal summation. So this would be if we have, here we've got a more detailed example, right? So here we're going to focus on presynaptic neuron A, okay? And the yellow cell is our postsynaptic neuron. So in temporal summation, we said before that that's adding together over time, right? So what we're talking about is the fact that if A fires just once, it is not enough to get us to threshold, right? So A is excitatory, creates an EPSP, an excitatory postsynaptic potential, but it's not one that's big enough to get us to threshold, okay? So when it fires individually, it's firing slowly over time with enough time to get back to the resting membrane potential in between. Nothing's going to happen for this cell. However, if A starts firing super fast, right, and we start to get those signals coming close together before they fade away back to rest. So here you can see A firing twice really close in a row. This means that that signal is going to add together over time. So that's our temporal summation. So A fired off neurotransmitter, gave us a little bit of a depolarization. And then before that depolarization faded away, it fired again, giving us a little more depolarization, and it got us to threshold. So we can see that action potential depolarizing up to positive 30, repolarizing down to its resting membrane potential, and doing that after hyperpolarization to about negative 85, so coming a little below, okay, before eventually we reset to our resting point. Okay. So that's our temporal summation, okay, versus our spatial summation. Now we're going to be thinking about this convergence, right? So we have our 3 presynaptic neurons. We have essentially 3 synapses. So we have the communication between A and this postsynaptic neuron. We have the communication between B and this postsynaptic neuron. Both A and B are excitatory, creating EPSPs, excitatory postsynaptic potentials. We also have C converging here. So C is an inhibitory connection. So C here creates an inhibitory postsynaptic potential. Okay. So in this case, we'd want to know what each of these does independently, right? So we can see that both A and B cause excitatory postsynaptic potentials. So we can see the depolarization from A. We can see the depolarization from B. And then we can see that if C fires, gives off its neurotransmitter, this causes a hyperpolarization. So that's our inhibitory postsynaptic potential from C. Okay. So when we're thinking about spatial summation, we're thinking about what happens if 2 of these fire at the same time instead of individually. So if A and B fire at the same time, that means we're going to have neurotransmitter coming in over here, creating an excitatory postsynaptic potential. And we have neurotransmitter coming in over here, more excitatory postsynaptic potential. So we'd add them together. And we can see that when both A and B fire at the same time, that gets us to threshold summing spatially, even though each one individually isn't enough to get us to threshold. So we can see that when A and B fire, we get an action potential. However, not all of these are excitatory, right? So if we have A and C fire, both of them release their neurotransmitters, right? But maybe we have our glutamate and our GABA fighting each other, right? So we have both excitatory and inhibitory neurotransmitters as options. So if A and C fire at the same time, A would usually cause this little depolarization. C on its own would usually cause this hyperpolarization. But if they fire off at the same time, nothing happens because we sum them over space, right? So as those postsynaptic potentials kind of drift and we measure at the axon hillock, overall there's no effect. Right. Questions about this? I know there's a little bit of confusion about the summation of the graded potentials when we talked about them at the beginning. I'm hoping that the more specifics that actually maybe may make them a little clearer, but I want to give you a chance to tell me if that's not.
For the bottom, something like when A and B go, that's been happening over separate times, like the first time they're happening about the same time, and then they're going again, like A and B, or is that just adding A and B?
And you were talking about the bottom one?
Yeah.
Okay. Yeah, so this is talking about just A at one point in time. So think of even labeled our x-axis is time, right? So yeah, so that's at one point in time, B at a separate point in time, so not at the same time, versus A and B together is when we have that spatial summation.
When they have to have the same
When they happen at the same time, you do add them up. So those two things go together. And maybe it'll help, the summation part, right? Summation really just means adding, right? So there are two ways we could add this together. Okay. Another piece of neural integration is frequency coding. So when we are thinking about frequency coding, what we're thinking about is frequency of action potentials. When we have more depolarization happening at the axon hillock, we might fire off action potentials more quickly. Okay? So the summation we've been talking about, right? So either over space or over time, but really we're just thinking about multiple signals getting us up to our threshold. If we get way above threshold at the axon hillock and it doesn't fade away, we would fire off more frequent action potentials, ultimately releasing more neurotransmitter at the opposite end of the neuron, and therefore creating a bigger postsynaptic potential, whether excitatory or inhibitory, in the next neuron. Actually, and I'm going to pull us back to the previous video. So essentially what we're saying kind of is a little bit of that is kind of what's happening in this temporal summation example, right? So if we have this slower frequency, we didn't add up to anything. When we have this faster frequency, we did add up to our threshold, releasing an action potential. And so you can imagine that if you have that axon hillock kind of staying depolarized longer, you would end up with this more frequent kind of signaling. So basically the way that we give a bigger signal to a cell in our system, rather than being able to change how big an action potential is, because we can't do that, right? They're always exactly the same size. What we can do is have more frequent or less frequent action potentials. More frequent ones means a bigger signal for the next cell, ultimately, because we have more neurotransmitter. So our last piece of this picture, this neural integration, I think is the most important piece, or at least the really new piece, is our presynaptic modulation. Okay. So we can regulate this communication between neurons kind of on a separate route than just directly acting on postsynaptic neurons, right? So in this image, we see a chain between a presynaptic neuron and a postsynaptic neuron, right? So we have some kind of communication happening from presynaptic neuron, action potential going down, releasing neurotransmitter, postsynaptic neuron doing its own thing, right? So we can change our signal to the postsynaptic neuron through some of this frequency coding, right? So more frequent or less frequent action potentials. But another thing that we can do is have a third neuron come in and have an effect directly on that presynaptic neuron right before it releases its neurotransmitter that affects how much neurotransmitter gets released to the postsynaptic neuron. Okay? So we can either cause more neurotransmitter to be released, or we can cause less neurotransmitter to be released, which means that we can have both facilitation and inhibition. So it's presynaptic because we're acting on the presynaptic neuron. Facilitation, anytime you facilitate something, you make it easier. Inhibition, inhibiting something is like suppressing it, right? So this is the role of the axo-axonic synapses. So to this point, we've mainly been talking about axodendritic or axosomatic connections, right, between the axon terminal and either the soma of the cell, which is just its body, so that's this main part, or a dendrite. We just don't typically draw them on, okay? But are slightly less frequent axo-axonic connections between the axon of a modulating neuron and a presynaptic neuron is how we get this modulating effect. So I've got a bunch of examples here. So what we see happening on this side here, we are looking at a postsynaptic neuron. Right, so our postsynaptic neuron here is neuron X. And what we're recording in this graph is the membrane potential of neuron X at that axon hillock. Okay. And we can see that coming in, we have presynaptic neuron D synapsing on the body of neuron X. We have presynaptic neuron C also synapsing on the body of neuron X. But over here on the right, neuron E is not actually sending its neurotransmitter to neuron X. It's sending them over here to the axon terminal of neuron C. So rather than directly affecting X, if E fires, it's going to be affecting how much neurotransmitter C releases. So when we see C fire alone, we see a slight depolarization. Okay. So that was this one. If we see D fire alone over here, slight depolarization. Okay. If E fires, absolutely nothing happens to X because E isn't touching X, right? So if E is firing on its own, X doesn't notice. It's when we have this combination between C and E that we see a difference, right? So if C and E fire at the same time, usually C would have a slight depolarizing effect on neuron X. And we can see how that's usually working. So here we see the amount of neurotransmitter usually released by C. If C and E fire at the same time, neuron E, acting on neuron C, increases that depolarization in the axon terminal, increases like how much calcium is coming in, all that good stuff, increases how much neurotransmitter C releases. So it's facilitating, making it easier for C to release neurotransmitter, which is why we have this larger effect when C and E fire together, which is enough to get to threshold, leading to an action potential. Because E only acts on C, if D and E fire at the same time, that's just the same as if D fired on its own, because E is not touching neuron C. So in this case, we'd refer to this modulating neuron E. So this would be our modulating neuron. We would say it's doing presynaptic facilitation because it made it easier for us to get to an action potential. It made C release more neurotransmitter. So you want to think about like that amount of neurotransmitter release when we're thinking about presynaptic facilitation versus inhibition. Questions about the facilitation example before we move to the other side and do the inhibition? All right. So inhibition works the same way, but opposite, right? So we see the same situation, except now we're recording neuron Y. We see both G and F have excitatory effects on Y, and we can see the depolarizations that they cause when they fire independently. So F a little bit, G a little bit. Neuron H does nothing if it fires on its own. Right, so here we see H. H is acting on F, so nothing happens if H fires. Now, when we think about the inhibition part, right, when F and H fire together, usually F is a little bit depolarizing, right? So if F fired on its own, we saw a little bit of depolarization, right? So we saw an excitatory postsynaptic potential, not getting us to threshold, but going in that direction. But now when we have F and H firing at the same time, we're not recording any difference in the membrane potential at all, right? So that's because the effect of H on F was inhibition, right? So this is presynaptic inhibition. So it happens before we get to the synapse that we're focusing on, right? So this would be the synapse we're focusing on, presynaptic, happening on that presynaptic neuron earlier than the synapse. So when H fires, it's decreasing the amount of neurotransmitter released, right? So F on its own is giving you a bunch of this excitatory neurotransmitter. When H fires, it suppresses, it inhibits F a little bit. So now we only have a tiny bit of neurotransmitter release. So that's how our presynaptic inhibition works. So just to keep things a little straight in your mind, want to make sure that you realize when we're talking about our postsynaptic potentials and the inhibition and excitation, we are focusing on this cell and whether it's depolarizing or hyperpolarizing, essentially, whether it's excited or inhibited. When we're thinking about modulation, we're not focusing on that postsynaptic cell. We're focusing on the amount of neurotransmitter released by the presynaptic cell, right? So we're on the opposite side of this equation. I'm just pointing that out because in the future, our neurotransmitters would be excitatory or they could be inhibitory in their effect on that postsynaptic cell. So it's not always going to be the case that presynaptic facilitation or presynaptic inhibition is going to cause one specific type of postsynaptic potential. So you don't want to be linking that with a specific direction. When we're thinking about this presynaptic facilitation and inhibition, we want to be focusing on amount of neurotransmitter. Amount of neurotransmitter released is the presynaptic facilitation or inhibition. our postsynaptic potentials. We're thinking about, does the charge here go up or down? All right. So we're going to imagine a situation now, thinking about this modulation. I'll draw it as we read it. Okay. So we've got our neuron 1, our presynaptic neuron. We've got our neuron 2, postsynaptic neuron. And then we're going to be thinking about neuron X. Okay. So neuron X is over here with an axo-axonic connection. 1, 2, and this would be X. What word would we use if modulator neuron X increases our amount of neurotransmitter that neuron 1 is releasing? Okay, look. Okay, yes, this is presynaptic facilitation. So when we have more neurotransmitter released by that presynaptic neuron, we made it easier for it to release neurotransmitter. That's facilitation, presynaptic facilitation. So we wouldn't need to know whether that neurotransmitter is depolarizing, hyperpolarizing, whatever, right? We're not thinking about the postsynaptic cell, just that presynaptic cell and how much neurotransmitter it releases. So when we think about our different types of synapses, we have now divided them a little bit in the types of effects that they have, right? So these axo-axonic connections that we've just been talking about as modulators are really going to be selecting just one synapse to act on, right? For the connection there, right? They're just going to be acting on that axon that they're touching. Okay, right? So they're acting just as modulators right here. For our axodendritic or axosomatic connections, I'll have to draw a slightly better neuron. Okay, so here's my postsynaptic neuron. The star points are the dendrites. The divots are going to be the soma, the cell body. Okay. Oh. Be lazy about this first one. All right. So our presynaptic neuron, an axodendritic connection, or an axosomatic connection, might also be diverging, affecting other neurons. This is not going to be particularly selective. It's not acting on just one little specific synapse. Neurons have many different shapes and can be really complex. We've been simplifying them an awful lot, just looking at this very typical type of neuron. But those axodendritic and axosomatic connections, we're going to be thinking about causing our excitatory or inhibitory postsynaptic potentials, just kind of overall as they release their neurotransmitter. Okay. Cool. So here we just have a summary of how our neurotransmitters work. So you can again see the full body of a neuron. You can see connections coming into it, all the labeled parts. And here we have that total flow of what's happening from the presynaptic neuron to the postsynaptic neuron as we're doing our signaling. Okay. So that's it for today. Next time we'll talk very briefly about a couple of other cell types that exist in our nervous system that are not neurons, and we'll do some review.